Hello, am I audible?
Yes, ma'am.
Yes,
Oh,
ma'am.
okay. Girls, just give me 5 minutes. I'm just coming in 5 minutes and then I'll start, okay? Okay. So today we're going to start and discuss about the anatomy of flowering plants. Now, you people, first of all, tell me what all things are there in this particular chapter. Like whatever you can recall from this chapter. Hmm? Am I audible, girls?
Yes, ma'am.
Hmm. Tell me what all you can recall from this particular chapter. What all things are there in this chapter?
Like from 11th.
Yes, this one, anatomy of flowering plants.
Ma'am, I didn't learn this chapter when I was in 11th. I guess it was deleted.
It was deleted.
I guess because I didn't learn this chapter.
It was not there in your syllabus at all.
Ma'am, it
No,
was
I don't
there,
think so.
but yeah, it was there, ma'am.
It was there now because like even after many like multiple topics have been deleted, but still it is like questions come from this also. Okay, anyway, it's all right now. Even if you did study or did not study, we're going to study about it right now. So anatomy, just like we learned in the previous class, morphology was the external appearance. Now anatomy is the internal structure. Now, in this chapter, we're going to talk about all the structures that are present inside the flowering plants, meaning the angiosperms. Now, we're going to see how the cells are organized and how they are present in the form of the tissue. Now we know that, okay, first of all, tell me what are the two divisions of the flowering plants? The angiosperms can be divided into angiosperms can be divided into dicots and monocots, right? Now we're going to see the whole entire anatomy with respect to these two only, because these are the divisions that we found. We'll find all the structures that are present in dicots and also the monocots. Now, within angiosperms, the monocots and dicots are also seen to be anatomically different. They're very different. So they're like, because of the way their cells are arranged, because of the places they are found in, and because of how their body structure is like, what is the thing that they procure and everything. Because of that, what happens is that their whole entire anatomy is also very diverse because of the adaptations. Now, more importantly, in terms of anatomy, we're going to talk about the variety of tissues that are found in the plant's body. Now, a tissue is a group of cells having a common origin and usually performing a common function. See, when many cells come together, they form a tissue. And why do they do so? So that there will be a division of labor. And by division of labor means that every single cell which is present inside the tissue, it would perform its own function. Now, a plant is made up of different kinds of tissues. Tissues are classified into two main groups, namely meristematic tissues and permanent tissues, based on whether the cells being formed are capable of dividing or not. See, meristematic tissue, or the term meristem, it denotes that the particular tissue or the particular cell will keep on dividing. It has the unique ability to divide. Talking about the permanent tissues, they are not capable of dividing, rather they are modified to perform other functions. Now, talking about the meristematic tissue, growth in plants is largely restricted to specialized regions of active cell division that are known as meristems. Plants have different kinds of meristem present in them. Now, the meristems which occur at the tip of the root and the shoots, they produce the primary tissues and are known as the apical meristem. Apical word means apex, apex means tip. So the ones that are present at the tip are known as the apical meristem. See, such as here, you can see here, root apical meristem, it's present here, and then there is the shoot apical meristematic area, or the shoot apical meristem. So it is present in the root and shoot both, so that the growth is supposed to happen. Now, root apical meristem occupies the tip of a root, while the shoot apical meristem occupies the distant most region of the stem axis. Now, one thing to remember here is that whether it is in the case of shoot or in the root, the meristem is not present here. Rather, it is present a little inside. Even here, the root is first of all protected by the root cap, and then only the meristem is found. Now, during the formation of leaves and elongation of stem, some cells left behind from shoot apical meristem constitute the axillary bud. So, now this tells us about the origin of axillary bud, and that is the shoot apical meristem. Now, such buds are present in the axil of the leaf. You remember axil of the leaf, the angle that is formed, that is the axil. And these axillary buds, they are responsible for the formation of a branch or a flower. Now, the meristems which occur between mature tissues is known as intercalary. They occur in between the mature tissues. Just 1 second, girls. Just 1 second. Hello, am I audible, girls?
Yes, ma'am.
Hello, am I audible?
Yes, ma'am.
Hello, 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 am I audible, girls?
Yes, ma'am, you're audible.
Okay, yes, yes. All right. So, yes, moving forward. Now, the intercalary meristem. See, these are the ones that occur between the mature tissues. So, in between the two mature tissues, there will be the presence of intercalary meristem. Again, the function is to divide only. Now, they occur in grasses and regenerate parts that are removed by the grazing herbivores. So, the regeneration in the case of the grasses, which is like primarily monocot, it is done with the help of intercalary meristem. Now, the apical meristem and the intercalary meristem, they come under the primary meristem. Why? Because they appear in the early life of a plant, meaning they are responsible for the formation of primary plant body. Now, moving forward, the meristem that occurs in the mature regions of roots and shoots of many plants, particularly those that produce the woody axis. See, as far as the primary meristems are concerned, they are responsible for the formation of, you can say, the length of the plant. Okay, they will form the length, they'll increase the length. Now, talking about this thing, the secondary meristem or the lateral meristem, what they do is that they actually increase the girth. They're not responsible for the length, rather they are responsible for the girth. Okay, they increase the thickness. Now, meristem that occurs in the mature regions of roots and shoots of many plants, particularly those that produce woody axis and appear later than primary meristem, is called as the secondary or the lateral meristem. They are cylindrical meristems. They like this, their structure is cylindrical in nature. Okay, they are cylindrical meristems. And what happens is that these are the ones that are responsible for the secondary growth and also the growth in the terms of girth. Girth means thickness. This much is clear, girls?
Yes, ma'am.
Any doubts up until now?
No, ma'am.
All right. Now moving forward, fascicular vascular cambium. Okay. The cambium which is present. So fascicular vascular cambium, interfascicular cambium. Okay. And then there is the core cambium. These are the ones that are the examples of the secondary meristems or the lateral meristems. Now, these are responsible for producing the secondary tissues. Following divisions of cells in both primary as well as secondary meristems, the newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed as permanent or mature cells, and it constitutes the permanent tissues. Now, during the formation of primary plant body, specific regions of apical meristem produces dermal tissues, ground tissues, and vascular tissue. See, dermal tissue, ground tissue, and vascular tissue. These are the three tissue systems of the plant body. The three basic or the three major tissue systems of the plant body is the dermal tissue system, ground tissue system, and then there is the vascular tissue system. What do you understand by dermal? What is the meaning of dermal? Dermis, dermal.
Skin?
Hmm?
Skin?
Skin, yes, outermost layering, correct, outermost layering. So, skin in terms of humans, but in the terms of plants, that is the outermost layer of their body. Okay, so remember this one thing, that the primary meristem or the primary plant body, more particularly the apical meristem, it would form the dermal tissue, the ground tissue, and the vascular tissue. This much is clear?
Yes, ma'am.
All right. Now moving forward, we'll talk about the permanent tissues. Now, the word permanent only is denoting the fact that they do not divide anymore. Now, the cells of the permanent tissues do not generally divide further. Permanent tissues having all cells similar in structure and function are known as simple permanent tissue. See, permanent tissue can further be divided into two types. One is simple and the other is complex. Now, when all the cells that are present in the tissue will be equal, same, then that type of tissue will be the simple tissue. But when different types of cells are present, we'll call them as complex tissues. Simple permanent tissue, complex permanent tissue. Now talking about the simple tissues. Now simple tissue is made up of only one type of cells. The various simple tissues in plants are parenchyma, collenchyma and sclerenchyma. Always remember this thing, okay? Because many questions can come from this part only: parenchyma, collenchyma and sclerenchyma. More importantly, their composition. Now, parenchyma forms the major component within organs. The cells of the parenchyma are generally isodimetric. All of the cells are similar and same in size. The cells can be either spherical in shape, oval, round, polygonal, or elongated in shape. Their walls are thin and made up of cellulose. So the composition of parenchyma is cellulosic. Always remember this thing. Okay? Cellulosic composition. All right. Moving forward, they may either be closely packed or have small intercellular spaces. Okay, they may, like for example, the cells may be arranged in such a way that there will be no spaces present in between them, or else they would have small intercellular, very small spaces present in them. Now, what is the function of parenchyma? It performs various functions like photosynthesis, storage, and secretion. Three functions of the parenchyma, very necessary. Okay. Girls, respond.
Yes, ma'am.
All right, moving forward. Okay. All right. Moving forward, we have collenchyma. Now, collenchyma occurs in layers below the epidermis in the dicotyledonous plants. See, right below the epidermis, there is collenchyma present. Now, it is found either as a homogeneous layer. Homogeneous means like uniform. Uniformly when it is present. So the collenchyma may either be present in a uniform layer or it can be present in several patches. It consists of cells which are much thickened at the corners due to the deposition of cellulose, hemicellulose, and pectin. Now, the composition of collenchyma is here. Cellulose is present, then hemicellulose and pectin. Three things are there. Now, they, like the collenchyma, it can be thickened at the corners mostly because of the deposition of extra stuff such as the pectin and hemicellulose. Now, collenchymatous cells may be oval, spherical, or polygonal and often contain chloroplast. If anything contains chloroplast, what does it mean? Or what does it denote? If anything, any cell, if it contains chloroplast or it's green in color, what does it denote?
Photosynthesis.
Very good. That it performs photosynthesis. Now, moving forward, these cells assimilate food when they contain chloroplasts. Intercellular spaces are absent. Now see, little intercellular spaces were present in parenchyma, but in the case of collenchyma, the intercellular spaces are absent. Now they provide mechanical support to the growing parts of the plant, such as the young stem and the petiole of the leaf. They provide mechanical support. Where do you think is this mechanical support coming from? Because of the presence of? Tell me. What do you think? I mean, according to you.
Stem.
No, no. Stem, no. We are talking about collenchyma. So they're saying that the collenchyma also performs the function of mechanical support. So how can it do? Like because of the presence of what thing does it make, provide mechanical support? What is that thing? See, it's right there. Think just a little bit.
Because the cells are quite thickened.
Because the cells are thickened and the presence of hemicellulose and pectin, it provides a thickening. And anything that has to provide support anywhere, it has to be solid itself first, right? So because of that. Now the last type of simple permanent tissue is the sclerenchyma. Now sclerenchyma consists of long, narrow cells with thick and lignified cell wall, having a few or numerous pits present in it. They are usually dead and without protoplast. Any cell that is dead, it means that the protoplast or the cytoplasm is not present in it. And the word lignified also denotes to its, like you can say, like it is dead. Okay, it has been lignified. Now, the sclerenchyma, they are usually dead and without protoplast. Now, on the basis of variation in form, structure, origin, and development, sclerenchyma may either be fibers or sclerites. The fibers are thick-walled, elongated, and pointed cells. This is a fiber. It is thick-walled, elongated, it is long and pointy. Generally, it occurs in groups in various parts of the plant. The sclerites are spherical, oval or cylindrical, highly thickened dead cells with very narrow cavities. The lumens are very narrow or very tiny. These are commonly found in the fruit walls of nuts, pulps of the fruits like guava, pear, and sapota, seed coats of legumes, and leaves of tea. What does it do? See, when we eat guava, apart from the seed, if you just eat the flesh of the guava, do you feel like it is a bit gritty? Like you would find like tiny, you'll feel like something tiny, hard structures are present in guava and even in pear. Have you ever felt that on your tongue? It feels like you're crunching it more. That crunching comes with because of the presence of sclerenchyma. Clear? So a very famous question that arises from the simple tissues is that which of the simple tissues are dead? So parenchyma is living, collenchyma is living, but sclerenchyma is the only dead tissue in the simple permanent tissue. Clear? Now again, since the sclerenchyma is lignified, so it provides mechanical support to the organs. Any doubts up until now?
No, ma'am.
Okay. Now the next is complex tissue. Now complex tissues, they will be made up of more than one type of cells. Here there will be no homogeneity. The cells will be very different from each other. Now the complex tissue are made up of more than one type of cells and these work together as a unit. Xylem and phloem constitute the complex tissues in plant. Now everyone must remember that the xylem and the phloem, they are the conducting tissues of the plant. Do you remember or not?
Yes, ma'am.
Right? They're responsible for the transportation of food and water.
Yes.
Very good. So the xylem functions as a conducting tissue for water and minerals. From where? So from the root to the stems and the leaves. It also provides mechanical support to the plant parts. It is composed of 4 different kinds of elements, namely tracheids, vessels, xylem fibers, and xylem parenchyma. Clear? So since it has so many components present which makes up one xylem, this is the reason why it is known as complex permanent tissue, because none of them are alike. Now gymnosperms lack vessels in their xylem. Gymnosperms do not have vessels in their xylem, just like how the normal ones would have the vessels present in the xylem, but gymnosperms do not. Now, talking about tracheids, Now, tracheids are elongated or tube-like cells with thick and lignified walls and tapering ends. It has tapering ends, just like how the muscles had tapering ends, so does the tracheids. Now, these are dead. Tracheids are dead and are without protoplasm. The inner layers of the cell walls have thickenings which vary in form. In flowering plants, tracheids and vessels are the main water transporting elements because they are always, you can say coordinated. Their activities are coordinated with each other, because of which they can transport water in the plant's body. Moving forward, the vessels. Now, vessel is a long cylindrical tube-like structure made up of many cells called as the vessel members, each with lignified walls. Again, the word lignin is coming, so it again means dead. Okay, so vessels are also dead. Tracheids are dead, vessels are dead. Now, vessels are also devoid of the protoplasm. It directly means that they are dead. Vessel members are interconnected through perforations in their common walls. The presence of vessels is a characteristic feature of angiosperms. Again, a very important point. The gymnosperms lack vessels, and the presence of vessels is a characteristic feature of the angiosperm. Now, then comes the xylem fibers. Now, they have highly thickened walls and obliterated central lumen. Very, you can say, very narrow or even negligible lumens. These may either be septate or aseptate. Septate means branched, aseptate means unbranched. Then comes xylem parenchyma. Now, these are living. The only component in the xylem that is living is the xylem parenchyma. Clear?
Yes, ma'am.
The only component that is living is the xylem parenchyma. They are living and thin walled, and their cells are made up of cellulose. They store food materials in the form of starch or fat and the other substances like tannins. Now, tannins are secondary metabolites. Do you know why secondary metabolites are formed in plants? Okay, first of all, tell me what is the difference between primary and secondary metabolite, girls? Is there any difference? Hmm? You can say, ma'am, we don't know. If you don't know, it's fine. If you don't have any idea, you can say that also. It's fine. No props. You're here to learn.
Ma'am, primary metabolites, they are essential for the growth of the plant.
Not actually growth, but actually the whole entire body structure only. Right? Because primary metabolites contain carbohydrates, proteins, which are very important for the plant's body. Right? Now, secondary metabolites are the ones that the plants do not need at all. They are made as a defense mechanism. Okay, so tannins, resins, alkaloids, okay, terpenoids, then there is saponins. These components, the resin, everything is made as a defense mechanism. So now you understand the difference between primary and secondary metabolites?
Yes, ma'am.
Hmm. All right. So so same thing, the tannins are present in the xylem parenchyma, and xylem parenchyma is mostly like like it primarily functions as the storing part of the xylem. So this is the function of each of the components of xylem. And now you people understand why the xylem is known as a complex tissue. Any doubts in xylem?
Ma'am, then how does the transport of water and mineral takes place in gymnosperms?
In a gymnosperm, see, the gymnosperm would have like you can say the tracheids, the xylem fibers and parenchyma. It just does not have the vessels, so it is not very, you can say, advanced type of transportation. In the case of gymnosperms, it is there, but not as efficient as the one that is present in angiosperms. Clear? Also, another thing is that in the case of gymnosperms, do you know like what is the habitat of gymnosperms? Like, where are gymnosperms mostly found?
Hilly areas.
In hilly areas. Mostly, see, uh, in gymnosperms, what happens is that their whole entire body, since they do not have the vessels, their major transportation process is done by the trachezoome. The rest of the components are there, only the vessel is absent. It's not that they do not have the xylem, it's just that they do not have the vessels. Clear? And this thing, the like since the gymnosperms, they do not have vessels most of the times in the question, like for assertion reason questions, they'll give the assertion as the gymnosperms lack trachees. है ना? So a person would see lacks and you're like, okay, हाँ चलो सही है, gymnosperm lack तो करता ही है कुछ ना कुछ. But again, the technicality is very important, क्योंकि एग्जाम के टाइम में ना बहुत बार ऐसा होता है कि we, like the obvious questions also, we sometimes do silly mistakes over there, isn't it? बहुत बार ऐसा लगता है कि अच्छा चलो ये तो सही है, ये तो सही है, and then later on we realize that okay, ये तो गलत हो गया है ना? So be like, more than anything, be very careful while reading the questions, क्योंकि अगर गलत ही पढ़ लिया तो फिर तो क्या ही फायदा है ना? So pay attention to that. Clear? All right.
Yes, ma'am.
Now, primary xylem is of two types. First is protoxylem, second is metaxylem. Now, the first formed primary xylem, they are the protoxylem. Proto means first. So, the xylem that is formed first is the protoxylem, and the xylem that is formed later on is the metaxylem. Now, in stems, now this is very, very important. You might think that ma'am underlines everything and says that it's important, but actually this is very important. See, in the case of stem, the protoxylem, which is the first form xylem, it will be present towards the center, towards the pith area in the center. So, and the metaxylem, which is the later form xylem, it will be present at the periphery. And this type of arrangement is known as end-arc. So end-arc arrangement is found in stems. Always remember this. End-arc arrangement is found in stems. Now, in the case of roots, the protoxylem would lie towards the periphery and the metaxylem would rise towards the pith or the center. And this condition is exarch and it is found in the roots. Don't get confused. Okay, end-arc stems, exarch roots. Always remember this thing. Now, this was all about the xylem. Now coming on to the phloem, which is the next type of complex permanent tissue. Now phloem transports food materials, usually from leaves to the other parts of the plant. Phloem in angiosperm is composed of sieve tube elements, companion cells, phloem parenchyma, and then there is the phloem fibers. Now again, यहाँ पे भी gymnosperm का discussion is there that they have the albuminous cells and sieve cells, but they lack sieve tubes and companion cells. So instead of companion cells, they have albuminous cells, right? And instead of sieve tubes, they would have sieve cells, but they would not have sieve tubes. Talking about the angiosperms, they'd have sieve tubes, companion cells, phloem parenchyma, and phloem fibers. Now, sieve tube elements, they are long, tube-like structures arranged longitudinally and are associated with the companion cells. Their end walls are perforated in a sieve-like manner. Perforated means a lot of holes are present, tiny holes are present, pores are present. Okay, that is perforation. Just say that thing is that our mothers use when we want to sieve the flour. Do you see that apparatus? Channi जिसे बोलते हैं. We put the flour onto
Yes,
it and then
ma'am.
we shake it and then the purified and the dust remains above. So that is a sieve, right? Same condition is of the sieve tube element. That is the name only. The tube has sieves present in it. Perforations. Okay. So now their end walls have such sieve-like manner perforations to form the sieve plates. Now a mature sieve element possesses a peripheral cytoplasm and a large vacuole, but it lacks a nucleus. Again, very, you can say, unique properties about the sieve elements is that they have a peripheral cytoplasm. Whenever we draw a cell, we draw the this thing. nucleus in the middle, the cytoplasm is here, and then we draw the vacuole also. But in this case, there is some peculiarity, such as the cytoplasm is peripheral in position, at the periphery. The major component or the major space is taken up by the vacuole, and the nucleus is absent in the sieve tubes. Like sieve tube is enucleated. Okay. So the thing that here means, okay, if it does not have the nucleus, then how will it work? How will it function? This is the reason why they have companion cells. Okay. Now, companion cells, like the name only suggests companion, right? A friend, a partner. So, companion cells, these are the ones that actually help or, support the sieve tubes. Now, the function of sieve tubes are controlled by the nucleus of the companion cells. Now they use the nucleus of the companion cells. Now, the companion cells are specialized parenchymatous cells, which are closely associated with sieve tube elements. Now, the sieve tube elements and companion cells are connected by pit fields present between their common longitudinal walls. The companion cells help in maintaining the pressure gradient in the sieve tubes. Apart from providing them their nucleus energy and everything, they also help in maintaining the pressure gradient. Now, phloem parenchyma is made up of elongated, tapering cylindrical cells which have dense cytoplasm and nucleus is present. The cell wall is composed of cellulose and has pits through which plasmodesmatal connections exist between the cells. The phloem parenchyma stores food material and other substances such as the resins, latex, and mucilage. Again, secondary metabolites are stored by the parenchyma of the phloem, so phloem parenchyma. Now, phloem parenchyma is absent in most of the monocots. Monocots do not have phloem parenchyma. Talking about the phloem fibers, they are also known as the bast fibers. They are made up of sclerenchymatous cells. If they are made up of sclerenchymatous cells, that means they are dead or alive?
Dead.
Dead.
Correct. They are dead. They are lignified. Protoplasm is absent in them. Now moving forward. Phloem fibers, these are generally absent in the primary phloem. In the first form phloem, the primary phloem do not have the phloem fibers, but are found in the secondary phloem, the phloem that is formed later on. Now, these are much elongated, unbranched, and have pointed needle-like apices. The cell wall of phloem fibers is quite thick. Now, at maturity, these fibers lose their protoplasm and become dead. Phloem fibers of jute, flax, sun hemp are commercially used. Like we know that the jute, everyone has almost jute bags in their homes, right? That jute comes from the phloem fiber only. It is a phloem fiber. Now, the first form primary phloem, it consists of narrow sieve tubes and is referred to as protophlloem, just like how the protoxylem is there. And the ones that are formed later on that have much bigger sieve tubes are known as the metaphlloem. So phloem and xylem, are they clear to you people?
Yes, ma'am.
Do you have any doubts?
No.
Do you feel like अब ऐसा लग रहा है कि मतलब बेमतलब में इस चैप्टर से नफरत थी या फिर नफरत बरकरार है?
अभी better feel हो रहा है.
बेटर फील हो रहा है. चलो, वेरी गुड, वेरी हैप्पी. अभी धीरे धीरे होपफुली और बेटर फील हो. चलो, नाउ वी हैव द टिश्यू सिस्टम. तो चलो सबसे पहले यही बताओ मेरे को, ये जो टिश्यू सिस्टम्स होते हैं, द डर्मल टिश्यू, वैस्कुलर टिश्यू और कंडक्टिंग टिश्यू, ये कौन बनाता है? हम्म? ऊपर बताया था ना, 3 टिश्यू सिस्टम्स कौन बनाता है? During the formation of primary plant body, specific regions of the apical meristem Apical meristem produces the dermal tissue, ground tissue and vascular tissue. तो कौन बनाता है? Apical meristem. ठीक है? चलो, moving forward. Now let us see the tissue system. Now, tissue systems are actually defined or explained with the, like in terms of their location, like where is it present in the body of the plant. So their structure and function would also be dependent upon the location. On the basis of their structure and location, there are three types of tissue systems: epidermal tissue system, ground or fundamental tissue system, and vascular or conducting tissue system. Talking about the epidermal tissue system, it forms the outermost covering of the whole plant body, and it comprises epidermal cells, stomata, and the epidermal appendages such as the trichomes and hairs. Now, one more thing. I don't know if it is mentioned later on or not. Take a hair. It has been mentioned. Okay. Now, the epidermis is the outermost layer of the plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. Obviously, epidermis interrupted kahin pe bhi nahi hota. It is a continuous layer. It is the outermost layer, and usually it is single layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall, and a large vacuole is present. So it has small amount of cytoplasm and it is parenchymatous in nature. If it is parenchymatous in nature, तो ये कौन से इससे बना होता है? What is the composition?
Cellulose.
Very good. Cellulose. Now, the outside of the epidermis is often covered with a waxy thick layer which is known as the cuticle. It prevents the loss of water. जैसे wax होता है ना, wax is surrounding the layer of the plant. That is a cuticle. Okay, but remember one very important thing, that cuticle is absent in roots. क्यों? Why? A cuticle is present all over the plant's body. जहाँ जहाँ epidermis है वहाँ वहाँ cuticle is present. But why is it absent in roots? ऐसा क्या खास है भाई कि roots में ही absent है? Hmm? See, what happens is that the cuticle, it is you can say waterproof. ठीक है? अब कोई भी चीज अगर waterproof होती है तो does it allow the water to come inside? Yes or no?
No.
No. अब भाई अगर रूट में क्यूटिकल प्रेजेंट होगा तो ये पानी को तो अंदर आने ही नहीं देगा. और रूट का फंक्शन क्या है?
Absorbing water.
Absorbing water. अब भाई क्यूटिकली अगर प्रेजेंट होगा तो the root can't absorb water. This is the reason why the cuticle is absent in roots. ठीक है? Clear?
Yes, ma'am.
Okay. थक गए क्या? Are you people tired?
No, ma'am.
No? अच्छा चलो, everyone switch on your videos. चलो थोड़ा सा मुंह भी देखें एक दूसरे का. I mean, let me see your faces. थोड़ी देर बाद बंद कर देना. शक्ल दिखा लो थोड़ी सी. हाँ ना, कुछ तो बोलो. I understand that the topic is very boring, but come on, students, respond तो करो भाई. कुछ तो respond करो. Girls, am I audible?
Yes, ma'am.
Yes, ma'am.
हाँ, what happened? नहीं करना on video. चलो करो, on करो, everyone, come on, quick, quick. करो, let me see, only I can see you. हम्म, भाई हाँ ना, कुछ तो बोल लो, कुछ तो बोलो क्लास में. हाँ, अब हुई ना बात. इरम, आप कहाँ हैं? आइए आइए, जल्दी से आइए. शो मी योर फेसेस. तो मेरे को लग रहा है, मालूम है कि मैं इधर बोल रही हूँ, यू पीपल आर स्लीपिंग. है. आई अंडरस्टैंड दैट द टॉपिक इज वेरी बैड, इवन आई डोंट लाइक दिस चैप्टर. चलो, स्विच ऑन योर वीडियो. सो यू पीपल हैव अंडरस्टूड क्यों क्यूटिकल नहीं प्रेजेंट होता है रूट्स में. हाँ, ठीक है. चलो, मूविंग फॉरवर्ड. Okay. Moving forward, stomata are structures that are present in the epidermis of the leaf. This is the place where stomata are present. Epidermis. Okay? Stomata regulate the process of transpiration and gaseous exchange. Each stoma is composed of two bean-shaped cells known as guard cells. Now, if you must remember that the guard cells are responsible for the opening and closing of stomata. याद है ये चीज? Do you people remember how does it regulate the opening and closing? Any idea? See, what happens is that, for example, if my two hands are the guard cells, okay? Now, if the guard cells are present like this, now what happens is that when the guard cells absorb water, they become swollen, and because of that, they open the stomatal pore like this. So, if it's a gaseous exchange, transpiration, everything takes place. But when the stomata loses the water, they become flaccid, so the stomatal pore closes like this. Okay, so this opening and closing of the stomata is regulated by the guard cells. Now, in the case of grasses, the guard cells are dumbbell shaped. Again, very important. See this. Normal guard cells, ऐसे होते हैं, ठीक है? And the guard cells of the what? The grasses. Now, the grasses are monocots. ठीक है? तो जो monocot के जो ये होते हैं, क्या बोलते हैं? Guard cells, they are dumbbell shaped. Now, the outer walls of guard cells away from the stomatal pore, they are thin, and the inner walls, meaning towards the stomatal pore, they are thickened. So, towards the inside, they will be thickened. This dark structure, here it will be thickened, and towards the outer side, it will not be thickened. Now, the guard cells, they possess the chloroplast and regulate the opening and the closing of stomata. Sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size and are known as subsidiary cells. Vicinity means in their near area. Now, the stomatal aperture, the guard cells, and the surrounding subsidiary cells, they all together form the stomatal aperture. Now, a question may come as to what are the constituents of the stomatal aperture. तो अब हमें पता चल गया, stomatal pore, guard cells, and subsidiary cells. And subsidiary cells of not other region, but the vicinity, meaning जहाँ उसके इर्द गिर्द. So, surrounding subsidiary cells, they would form the stomatal aperture. Now, the cells of epidermis, they bear a number of hairs. Now, from the epidermis only, they are saying hairs. Why? Because it is countable. Now, the root hairs are unicellular elongation of the epidermal cells and help absorption of water and minerals from the soil. On the stem, the stem hairs are also there. The epidermal hairs of the stem are known as trichomes. Now, remember one thing, that the trichomes are multicellular, the root hairs will be unicellular. So this is again a technicality that you must remember. Now, the trichomes may be branched or unbranched and soft or stiff. They may even be secretory. Now, the trichomes help in preventing water loss due to transpiration. Trichomes are extremely important because they're sort of like hairs present on the surface of the stem, which will allow the stem to actually not lose a lot of water. So the epidermal tissue system is clear?
Yes, ma'am.
All right. Moving forward, we have the ground tissue system. Now, all tissues except the epidermal and the vascular bundles will make the ground tissue. It consists of simple tissues such as parenchyma, collenchyma, sclerenchyma. Parenchymatous cells are present in the cortex, pericycle, pith, and medullary different regions of the plant, and more importantly, different regions of the primary stem and the roots. In leaves, the ground tissue consists of thin-walled chloroplasts containing cells and is known as mesophyll. So the ground tissue of the leaf is mesophyll cells, and the ground tissue, see, what is the meaning of ground? Ground means the basic tissue that forms the body, the structure. That is the simple permanent tissues. Now comes the vascular tissue. Now, vascular tissues, these are the conducting tissues. They are responsible for the conduction. Now, the vascular system consists of complex tissues, the phloem and the xylem. The xylem and phloem together constitute vascular bundles. They are known as vascular bundles. In dicot stem, cambium is present in between the phloem and xylem. The presence of cambium actually helps them to perform the secondary growth, because the cambium possess the ability to form secondary xylem and phloem. And those kind of vascular bundles that have the cambium present, they will be known as the open vascular bundles. Moving forward, in the case of monocots, cambium is not present, and since they are known as closed vascular bundles. Now, when xylem and phloem within a vascular bundle are arranged in an alternate manner on different radii, the arrangement is known as radial, like when they are present on different radii. And this happens in the case of roots. So the arrangement of vascular bundles in the case of roots is radial. In conjoint type of vascular bundle, the xylem and phloem are situated on the same radii, and such are found in the stem and leaves. So stem and leaves have conjoint vascular bundles. The roots have radial vascular bundles. The conjoint vascular bundles usually have the phloem located only on the outer side of the xylem. Again, Endark situation is there. Moving forward, now we'll do a comparative analysis of dicots and monocot plants, and that is all that there is to know in this particular chapter. Okay.
Ma'am,
Hmm.
ma'am, we have from secondary growth it is deleted. 6.4.
Yes, secondary growth is deleted. I know that. This dicot root, dicot stem, dicot this thing, monocot root, monocot stem. These things are there, right? Up, like from here,
Yes, ma'am.
everything is deleted, isn't it?
Yes. Yes.
Okay. Cork, cambium, and all, everything is deleted.
Yes, called cambium is deleted.
Okay. All right. Now we're going to talk about the different anatomies that are found. Okay. So let's talk about the dicot root. Now, dicot root, the outermost layer will be the epidermis, and the epidermis will give rise to the unicellular root hairs for better absorption. The cortex consists of several layers of thin-walled parenchyma cells within the intercellular spaces. Parenchymatous cortex is there. The innermost layer of the cortex is known as endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells, they have a deposition of impermeable waxy material, suberin, in the form of Casparian strips. These are the waxy materials that are present in the endodermal area. Next to the endodermis lies a few layers of thick-walled parenchymatous cells that are known as pericycle. So outside is epidermis, which gives rise to root hair. Then there is cortex. Inner to cortex is endodermis. Now the endodermis gives rise to the pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in the cells of the pericycle. Now talking about the pith is small or inconspicuous. The pith is very small. The parenchymatous cells which lie between the xylem and the phloem are conjunctive tissue. Conjunctive tissues. Now they are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and the phloem. All tissues on the inner side of the endodermis, such as the pericycle, vascular bundle, and the pith, constitutes the stele. So, pericycle, vascular bundle, and the pith makes the stele. Remember that because they ask about the constituents present in a stele. So, you should know. Now, moving forward, there is the monocot root. Now, the monocot root anatomy is very similar to the dicot. It has epidermis, cortex, endodermis, pericycle, vascular bundles, and pith. As compared to the dicot root, which have fewer xylem bundles, there are usually more. Such as we saw here in this case, it had 2 to 4 xylem and phloem. Here, more than 6. Polyarch condition is found in monocot root. Pith is large and well developed. In the case of dicot, it was small and inconspicuous. Monocotyledonous roots do not undergo any secondary growth, so they are closed. Closed means cambium is absent. Now moving forward, we have dicot stem. Now the transverse section of a typical young dicot stem shows that again, epidermis is the outermost layer of the stem, covered with a thin layer of cuticle. Now the epidermis would give rise to the epidermal hair, that is the trichome, which is multicellular. Now the cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. There is the outermost hypodermis, which consists of collenchymatous cells. Just below the epidermis, hypodermis is present. Later on, and obviously since collenchyma is there, it would provide mechanical strength to the young stem. Then there is cortical layers below the hypodermis, and it consists of rounded, thin-walled parenchymatous cells with conspicuous intercellular spaces. Talking about the innermost layer is the endodermis. Now the cells of the endodermis, they are rich in starch grains, and the layer is also known as starch sheath. So again, a characteristic feature of dicot stem is the presence of starch sheath. Talking about the pericycle, so the pericycle is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundles, there are a few layers of radially placed parenchymatous cells which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundles is a characteristic of dicot stem, a ring-like arrangement. Each vascular bundle is conjoint, open, and end dark because we're talking about the stem, not the root. Now, a large number of rounded parenchymatous cells with large intercellular spaces occupy the central portion and makes the pith. So the pith of the this thing dicot stem is parenchymatous. Now coming on to the monocot stem. Now the monocot stem has a sclerenchymatous hypodermis. The hypodermis is made up of sclerenchyma. A large number of scattered vascular bundles are there. In the case of dicot, it was a ring. Here, it will be scattered. Now, each of those are surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. The vascular bundles are conjoint and closed, meaning cambium is absent. Peripheral vascular bundles are generally smaller than the central located ones. The phloem parenchyma is absent in monocot stem, and water containing cavities are present within the vascular bundles. So like a little difference and more technicalities as compared to the dicot stem is there. Now talking about the dorsiventral leaf or the dicot leaf. Now, the vertical section of a dorsiventral leaf through the lamella shows 3 parts: epidermis, mesophyll, and the vascular system. The epidermis, which covers the upper surface, that is the adaxial epidermis, and the lower surface, that is abaxial epidermis of the leaf, has a conspicuous cuticle present. Now, the leaves have conspicuous cuticle, not a very evident one, but the one that will be enough to help it. Now, the abaxial surface. Abaxial means the lower surface or the backside. It has more stomata than the adaxial, that is the front or the upper surface. Sometimes in some cases, the adaxial surface, meaning the upper surface, may even lack stomata. Now tell me why. Why is that the case? बताओ, लीफ के लोअर सरफेस पे why should there be more stomata as compared to the upper surface? Girls, say answer. So that excessive water loss does not take place, because stomata say transpiration and gaseous exchange hota hai upper surface पे, if it will be present in more number, so it will receive more air, because of which more and more water loss and gaseous exchange will take place. That is the reason why lower surface has more stomata as compared to the upper surface. Girls, are you there in the class or not?
Yes, ma'am.
Okay. Now the tissue between the upper and the lower epidermis is known as the mesophyll. Mesophyll, which possess chloroplast and carry out photosynthesis, is made up of parenchyma. It has two types of cells, like the mesophyll is made up of two types of cells, palisade parenchyma, and then there is spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cells and extends to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes the vascular bundles, which can be seen in the veins and the midrib. The size of the vascular bundle are dependent on the size of the veins. The veins vary in thickness in the reticulate venation of the dicot leaves. Vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. So, mesophyll cells, bundle sheath cells, these two types of cells are found in the leaf of the dicot, and the dicot leaves have the reticulate venation. Now, talking about the monocot leaves, they are isobilateral leaves, and they're almost similar, but they have their own unique properties. First of all is that in this case, isobilateral means that the stomata are present on both the surface of the epidermis. And also the unique property is that the mesophyll of the monocot leaf, they are not differentiated into palisade and spongy parenchyma. I mean, it is made up of parenchyma, but it is not differentiated into spongy and palisade type of parenchyma. In the case of grasses, certain adaxial epidermal cells along the veins modify themselves into large, empty, colorless cells. These are known as the bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, they make the leaves curl inwards to minimize the water loss. Same way how the guard cells perform. Now we know that the monocots, they have parallel venation, and leaves is reflected in the near similar sizes of vascular bundles. Since their leaves are like all of them have the same kind of veins present, so that is why it is the case. And this like these are all the certain exceptions that are there for the monocot leaf. Now we have completed this chapter. Now tell me if you have any doubts, anything that there is to ask. Is the chapter clear, girls?
Yes, ma'am.
Any doubts?
No, ma'am.
No? All right. Okay, so we have completed this chapter. अब भाई देख लो, this chapter requires a lot of reading because यहाँ पे there are like multiple technicalities, isn't it? कितना सारा variation है in terms of the cells that are present, in terms of this thing, the cells arrangement and everything. So you have to pay close attention to that. Okay? Any doubts?
No, ma'am.
All right. So now the next chapter that we will be starting will be Animal Kingdom. Okay. Now this is a very interesting chapter. You will love it. Animal Kingdom. Anatomy समझ में आ गया ना?
हाँ, ma'am.
Sure?
Yes, ma'am.
चलो, good. Okay, now we'll move on to Animal Kingdom. Let's take 5 minutes break and उसके बाद फिर Animal Kingdom start करते हैं. I'll use my notes. Do you like this chapter? ये तो अच्छा
Yes,
chapter
ma'am.
है ना? ये
It's
तो
good, but examples थोड़े ज़्यादा हैं.
अच्छा. हाँ, examples आते हैं. चलो, we'll discuss everything about it 5 मिनट के बाद. ठीक है, बस मैं अपने गले को रेस्ट दे दूँ. Only 5 minutes, okay? You people can also go stretch. 5 मिनट बाद आ जाओ.